So it gives me great pleasure to introduce to you Dr. Judith Worthington, Principal Clinical Scientist at the Manchester Renal Transplant Unit.
Good morning, everybody. Thank you very much for the opportunity to talk to you today, just to tell you a little bit about what the lab does and how it supports living donation. So I can tell you a little bit about the discipline that I work in first of all. So I work in histocompatibility and immunogenetics, which is the H&I bit that you can see there. And histocompatibility is Greek for tissue, and immunogenetics means the study of genes. We're a little bit different than most laboratory pathology services in that we're not in every hospital. So we're not like biochemistry and hematology where we're in every hospital. There are only actually 27 labs in the UK and Ireland, as shown on this slide. And the North is quite a good example of the division of the labs, because about half of the H&I labs are based in the NHS blood and transplant, so the blood transfusion service. So in this case, Newcastle and Sheffield are that. And then the other half of the labs are based within transplant units like myself. We're based within the Manchester Royal Infirmary with the kidney transplant unit here. So your HLA or tissue type, HLA stands for human leukocyte antigen, and it can be thought of as like a barcode, the same as you get on products in a supermarket. So the barcode is present on all the cells in your body, and it's completely unique to you unless you happen to have an identical twin. And tissue typing is a series of lab-based tests using DNA that works out what your barcode is. So the blood sample that we usually use is usually used to determine what your tissue type is. So that's quite easily taken. But because HLA is expressed on all the cells in your body, we can actually use a variety of other ways, other sources. So we can use buccal swabs, mouth swabs, we can use blood spots, and we've even been able to tissue type from somebody's toenails. So we can use a variety of sources to get tissue type. So what does a tissue type actually look like? It's a complicated string of letters, symbols, numbers, colons, and everything. So this is actually my tissue type that you're looking at here. Everybody who starts in the lab is tissue typed because we're all a bit geeky and we like to know if anybody's got a really weird and unusual tissue type. As I say, it's a lot of numbers, colons and stars and everything else. But what we're mainly concerned about for renal transplantation is looking at 3 particular genes. So HLA-A, HLA-B and HLA-DR. And everybody has 2 A's, 2 B's and 2 DRs. And I'll explain why that is in just a second. So you're more than welcome to ask your clinicians for a copy of your HLA type, but as I say, it's just basically a list of numbers, colons and stars. So your tissue type is determined genetically. So you get half your tissue type from your mum and half your tissue type from your dad, which is why everybody has 2 A's, 2 B's and 2 DRs, because you get one of each from mum and dad. And these are inherited together in strings or haplotypes. So if we look at this famous family that we've got here, we've got Homer and Marge and their tissue types, and we've got their 3 children, Maggie, Bart, and so what we're going to do is you can see that Bart has inherited the A2B44DR4 from Homer, and he's inherited the A11, B55 and DR14 from Marge. Whereas Maggie has inherited the A3, B15, DR7 from Homer and the A1, B8, DR3 from Marge. So in any mum-dad combination, there are 4 combinations that the children could possibly be, because they could inherit this the A2, B44, DR4, or they could have inherited this one. And also, so that when you look at the children, this means that you've got a 1 in 4 tissue type, a 1 in 4 chance of being exactly the same tissue type as your brother or sister. which is the case that we've got here, or you've also got a 1 in 4 chance that you'll have absolutely nothing in common with your brother or sister as well. So the fact that you're more likely to share part or some of your tissue type with your family is the premise behind living donation. So if we go back to the barcode analogy, so if all your cells have a barcode on them, the scanner part of this analogy is your immune system. So your immune system scans every cell as it goes past it, checking the barcode, making sure that it belongs to you. And if it spots a barcode that isn't yours, it will flag it as being foreign. And then your immune system will work to remove that cell from your body. So part of the way that your immune system deals with foreign barcodes is to use antibodies. So antibodies are proteins that are found in your blood, and they bind to cells that are different from your own and alert the immune system. So they act as flags to show that the cell is different from what the rest of your body is. I think in this COVID era, everybody's heard about antibodies and how important they are in fighting viruses and infection. But unfortunately, they're also very, very good at flagging transplanted organs as being foreign and causing your immune system to attack those transplanted organs. So I said the blood sample that we use to test for, we use an EDTA blood sample to do your tissue type, and we use a clotted blood sample to test for HLA specific antibodies. So if you came to us for initial tissue typing, those are the 2 samples that we would take from you, 5 ml EDTA and 10 ml clotted blood sample. So what causes HLA specific antibodies? So they're called HLA specific antibodies because they're targeted against specific HLA or tissue types. You can't catch these, you can't be inoculated in them with them, or the only way that you can produce HLA specific antibodies is when your immune system sees somebody else's tissue type. So there are only 3 ways that that can happen. So the first one is pregnancy. So as I explained, you get half your tissue type from mum and half your tissue type from dad. So when mum's pregnant, half of the baby is dad's tissue type. So her immune system is exposed to dad's part of the baby during pregnancy. The other way is blood transfusions, although this is much less of a problem in this era because most blood transfusion units are filtered for white cells to remove them. And the third way is through transplant, because there are actually over 200 million different tissue types. So the chance of you getting a perfectly matched kidney is very slim. So if you've had a previous transplant, whether it's a kidney or another organ, there's a chance that you will have been exposed to somebody else's, or part of somebody else's tissue type by transplant. And the more pregnancies, transfusions and transplants a patient has, the more likely they are to produce antibodies. So one term that you might hear used by the clinicians is your calculated reaction frequency, or some clinicians still use the term panel reactive antibodies. This is a percentage that gives an indication of how many antibodies you've got. So if you look at the first table, this is a list of different HLA-A tissue types, and the patient that we're looking at has no antibodies, so all of those tissue types are acceptable for them. So then they are to have 0% antibodies. If you look at the second table, it's the same list of HLA antigens, but this patient has developed antibodies to all of the ones marked in red. So they have antibodies to 19 out of the 21 different tissue types, which works out as a percentage of 90%. So they have 90% antibodies. So once we detect an antibody in your sample, we would consider that to be unacceptable in a transplant for you. So we wouldn't consider any donors that had that tissue type. So this first patient could be considered against all donors, and this patient will only be considered against 10% of donors because the other 90% are unacceptable for them. Now, having said that you can only get HLA-specific antibodies 3 ways: pregnancy, transplant and transfusion, we do ask patients to send us samples on a regular basis. And this is because the amount of antibody can change over time. So the tests that we do to determine your antibody status actually give us a numerical output similar to what you might see for your hemoglobin or your serum creatinine. So this graph on this slide just shows that how the levels change over time, the orange line, and the blue line is the test cutoff. So when this fictional patient initially came to us in 2015, the antibody was there, but below our test cutoff because it was a very low level. And then over time, the level of the antibody is increased and we can detect the antibody in the serum. And then again, later on from 2018 onwards, the level starts to drop down again and then once again drops below the test cutoff. So when we test you for antibodies, it's only really a snapshot in time, and it's important that we have this longitudinal data from anybody on the transplant list so that we can see how your profile changes. And the reason that your profile might change is that whilst you might not have had a transplant, a transfusion or a pregnancy, your immune system has a memory. So you could experience a random event that stimulates your immune system back into action again, and it can, as a byproduct of that, can start producing these HLA-specific antibodies again. So the sorts of random events that I'm talking about are if you have an infection, peritonitis, if you catch a bad cold, or if you have a vaccination, because vaccination stimulates your immune response to respond. So that's why it's very important that we collect regular samples from patients. So the third test that we do in the laboratory is a crossmatch test. So we type patients to work out what their tissue types are, we screen them for antibodies, and the final test is a crossmatch test. And when I first started working in the lab, it was described to me as like doing a transplant in a test tube. If it doesn't work in the test tube, then you're not going to go ahead and do the kidney. So we don't actually do them in test tubes. We do them in these plastic trays that you can see as the background to the slide, very small wells, and we put a small amount of patient serum in these wells, and then we add a small amount of donor cells. And if the patient's serum has antibodies that is specific to that particular donor's tissue type, the antibodies will bind to the cells and then kill them, so they'll burst open. So then we add fluorescent dyes, and if the antibody has bound to the cell and killed the cell, the dyes will go into the cell, and down the microscope they're this red-orange colour that you can see in the positive crossmatch well. If the patient's serum has no antibodies in it that are specific for that particular donor, the cells don't burst open, they're not killed, they stay intact, and the dyes coat the outside of the cells, and down the microscope they're this green colour, so a very negative. This is termed a negative reaction. So a positive crossmatch means you have antibodies that would attack the kidney and therefore the transplant can't go ahead. A negative crossmatch means that you have no antibodies that would attack the kidney and therefore the transplant can go ahead. And this test is the crucial pre-transplant test that is done. So for deceased donors, this would be done in the acute situation and would take about 4 hours for us to complete the test. For living donors, it's done in a more planned process, and it's done the week preceding the transplant, the living donor transplant. So those are the tests that we do in the lab. We type people to get them on the transplant list. We type potential deceased donors, we type potential living donors to assess how compatible they are. We screen patients regularly for antibodies, and before anybody has a transplant, they will have a crossmatch test. I just want to go over 2 more terms with you just to set the background, to just explain how we fit into the living donor programme. So the first one is ABO blood groups. Now everybody's familiar with the fact that they have an ABO blood group, and most people do know what their blood group is. this table here shows what is termed compatible and what isn't. So if you have a donor whose blood group is O, that donor can donate to any recipient, irrespective of what their blood group is. By the same token, if you have a recipient is blood group AB, then they can receive a kidney from any blood group donor, again, irrespective of what blood group is. So O is called the universal donor and AB is called the universal recipient. Other than that, each patients will have antibodies which will stop them being able to accept a different ABO blood group. So a recipient who is O can only have blood group O kidneys because they have antibodies that would attack the donor kidney otherwise, and vice versa. So this is the ABO compatibility. So the other thing to think about is HLA compatibility. So there's a lot to talk about HLA matching, but it's actually mismatching that's important. So if we go back to our family again, this is Bart's tissue type, and these are his potential donors. And we can have a look at who's going to be the most compatible donor with him from a HLA point of view. What's important is what the donor's got that the recipient hasn't, because that's what the recipient would mount the immune response to. So if we look at the first one, Bart is A2 and 3. It's the 3 that he's got that, Homer's A3, which Bart hasn't got. So this is a mismatch. 1544, it's the 15, so it's the 15 that's the mismatch. And DR4 and 7, it's the 7 that's the mismatch. And these are expressed as 3 number codes. So this would be a 111, 3 mismatches out of 6. Whereas here we've got a 000, so Lisa would actually be HLA identical for Bart. And Maggie is 2 mismatches at A, 2 mismatches at B, and 2 mismatches at DR, so she's a complete mismatch for Bart. So just to, sorry, there's a little bit of crossover with Mr. Campbell's slides here, but just to talk about the types of living donation. So we look at potential donors who are genetically or emotionally related to the recipient, so such as a parent's child, brother to sister, or friends to friends. So whilst you've got the best chance of finding a genetic match in your family, it's not limited to looking at family members. It can be people that you know and have been friends with for a long time. The other type of donation is directed altruistic donation, where you're donating to a specific individual, but you don't have that genetic or emotional relationship, such as a work colleague donating to a colleague's child, or we've had instances where members of church groups have come forward to donate to an individual. And a non-directed altruistic is where a person donates an organ to an unknown recipient and there's no contact between the donor and recipient. So where do we fit in the lab when we're doing all the testing? So the testing that we do is the same as we do for deceased donors, it's just in a different order. So when a potential donor comes forward, as Mr. Campbell said, they will fill in a health questionnaire and have an initial consultation with the living donor nurses, and then they will be consented to take bloods for HLA typing and ABO typing. So from the potential donor, we need to get bloods for typing and a copy of their ABO report. From the potential recipient, we need to do the same thing. We need to type them to see what their tissue type is. We need to screen them for antibodies. We need a copy of their ABO report, and we also need to know their immunological history. So we need to know, have they had any pregnancies, transfusions, transplants, so that we can adequately assess them. And then we put all this information together and we produce an interpretive matching report. And as part of that interpretive matching report, we will include what transplant options there are for each donor and recipient pair. So the possible options, you could be completely ABO and HLA compatible, and you'd be suitable to directly donate to each other. It could be that we have a concern that you may have some antibodies that may be donor directed, and we want to assess that further. So we would then say we recommend that the next thing we do is cross-match this particular donor and recipient pair. It may be that we say you are not ABO compatible or you're not HLA compatible, and there is an option to do antibody incompatible transplantations in the UK, but it's tended to be surpassed now by registration in the National Sharing Scheme, which Mr. Campbell also talked about. Whichever option is decided on, whether it's direct donation or whether it's a transplant through the sharing scheme, as I said, the one thing that we do need to do is the final pre-transplant cross-match. Now, again, I know Mr. Campbell touched on this, but this is just a bit of a further explanation about the paired pooled scheme. As Mr. Campbell says, once you're registered in the sharing scheme, there are 4 runs a year, and the algorithm is set up to maximise the number of potential matches and prioritise the patients who have waited longest. So there are various different exchanges in the sharing scheme. So the most simple exchange, and the way that the scheme started out, is a 2-way exchange. So if we imagine that this is donor 1 and recipient 1 who can't donate to each other because they're ABO incompatible. However, donor 1 can donate to recipient 2, and donor 2 can donate to recipient 1. So it's just a simple exchange there. The scheme then expanded further after about 18 months to include 3-way exchanges, exactly the same principle. It's just that there are 3 donor and recipient pairs who can't donate to each other, but they can. The donor 1 and donor recipient 1 can't donate to each other, but they can donate within the 3-way exchange. And then more recently, they've incorporated altruistic donors into the sharing scheme. So the altruistic donor would donate to 1 recipient. Their donor, their coordinating donor, would then donate to the waiting list, and that's called a short donor chain. Or you can have long donor chains that include more donor and recipients, ultimately ending with your donor donating to the transplant waiting list. NHSBT have just very recently launched this website, which is a patient information website. There is a living donation page which explains much better than I have about the sharing scheme. And there's also lots of other information on that webpage, so I would recommend you looking at that if you're looking for further information. It was done in cooperation with patient groups and donor nurses, so it's very informative. And that's me. If there's any questions, I'd be happy to take them now, or we could wait for later.
Thank you, Judith, for your very thorough and comprehensive explanation of what can be quite hard for patients to understand, particularly the matching, and also for explaining so carefully about the UK living donor shared scheme.
I say the website's much better than me at doing that.
So is that back up and running normally now?
It is, yes. So obviously it was suspended during the pandemic, but we're back on track now. There's been a run in July and October this year, and next year is scheduled to proceed as normal. Excellent.
I think Tunde has been diligently answering quite a lot of the surgical questions. Just checking. No, there's nothing else in the Q&A. Don't know if we haven't had any in the chat. Yeah.
So I've either blinded you all with science, or I was so clear that you don't have any questions. I'll go with.
Personally,
so clear.
You demystified the 111 and the 000, the 222 for me, because I've seen different and heard different numbers Was it correct that they changed the description of the matching,
Yeah, I think
the cross matching?
I think sometimes the problem is that it's talked about matching, but it's actually mismatching.
Mismatching, yeah. And that's very, when you see that on your clinic letter and it says mismatch, it seems almost a contradictory term in many ways. Thank you. Let me just check. Susan's been posting links, by the way, in the chat, and we'll continue to do that for anyone who is perhaps contemplating donating, or a potential recipient in the audience who is wanting to give an answer to a potential donor, a family member, or a friend about the process involved.
There is somebody in the chat, Tess, who's posted a little message, and this lady says that her workup to donation took 15 months because the process is so thorough, and the extra investigations that she needed. But it all worked out in the end, and her advice was don't give up. So thank you for that.
Thank you. And Judith, there is a question that has just popped up. Do the chances of a second transplant reduce because of antibodies created during the first?
Unless you receive a perfectly matched kidney, it's likely that you will produce some antibodies, and those antibodies will influence your chance of future transplantation, but it all depends what those antibodies are. So if you receive a well-matched graft, you've had limited exposure to other HLA antigens, you may produce a small number of antibodies, so a low percentage, as we explained. If you are exposed to multiple mismatches, and some of those are very common, then you may end up with a large CRF percentage, panel reactive antibody percentage. So the panel reactive antibody, or the CRF, is an indication of the percentage of the last 10,000 UK donors that would not be suitable for you. So if you have a panel reactive antibody, sorry, I hate that term, but it's the one that's most used. We use calculated reaction frequency. So if you have a calculated reaction frequency of 20%, it means only 20% of donors wouldn't be suitable for you. But if you have a calculated reaction frequency of 90%, then 90% of donors wouldn't be suitable for you. But the influence purely depends on what you're exposed to in that first transplant. So if your first transplant is well matched, it shouldn't significantly impact your chances of a second transplant.
Thank you. I just had one question about the effect of COVID or COVID vaccination on, because you did mention that that of course can influence your immune response.
Yeah, there was a lot of concern, obviously, when this first all started, that we'd see a lot of antibody production in patients as a result of COVID. But that hasn't, in our experience, been the case. When the programme was suspended in Manchester, before we reactivated the programme, we screened everybody on the transplant list to make sure that they hadn't produced any new antibodies. And we didn't really see a significant increase in antibody production. So I think that hasn't been the case. It was a concern, but I don't really think that has been a problem.
So is that in people that had COVID?
Yes, we've been keeping a note when we've known that somebody's had COVID and checked whether it's had any influence on their HLA antibody status, and it doesn't seem to have done.
Okay.
Thank you.